Is this on?
Is this thing on?
You ready to go?
Which side did I part my hair on? Question mark?
Okay.
Red leather, yellow leather, red leather, yellow leather.
Every morning.
No, I haven't done it in months.
Here we go. Again. Ooh, there's time. Welcome, welcome.
Good morning.
Hello.
Hello.
We are already recording.
We are.
Yeah. What's up with that?
No.
No. What did you do? What did you say?
Thanks, 5440. Those are 3 chords in a very pleasing arrangement.
Wow. Isn't that kind of what music is? Chords and pleasing arrangement?
Yeah, but 3? That's
Okay.
not many chords.
Wonderful. We hope that you
And a flute. That's the first flute of the course.
have had a wonderful weekend.
Yes, did you get to shovel? That was the big highlight for our family this weekend,
Yeah.
that there were children shoveling.
Doing work as they should.
Yeah. Now, off to the coal mine. We get up
Yeah,
in the
wonderful.
black.
Okay, please finish the fish, because you're doing a great job. I'm really digging the kind of like teal and red, like that's like super intense. Yay, I love it. Okay.
Isn't it lovely?
It's lovely. Okay. Yes, we do have a topic test coming up on Friday. The last topic test. We hope that as far as tests go, you have been enjoying them. I know tests, they suck. But anyway, we hope that these ones are not as bad as other tests you have had in the past. We will be around on Wednesday during regular class time if you want to drop in and ask us any questions or anything like that. We're just going to do like a extended kind of student hour for drop-in on Wednesday. So if
Do you want to let people propose things?
to talk about,
Yeah.
oh yeah, throw some topics in the chat, and we can totally like be ready with slides or opinions.
Or stories.
Or stories. If you want to hear stories, we have stories. But if you want to talk about the topic test, then we can definitely, it's your time.
When is the Squirrel Life Project due?
I feel like, I don't know, but I feel like,
Where would you look?
yeah, that it's on CourseLink in the Squirrel Life Project content section. I would do it as soon as possible.
Can we take a moment to say hello to everyone beside you at Creelman's?
Hello! Are you at Creelman? That's like the best place to eat. Oh, I'm thinking about Creelman Hall now. Okay. Because it's like breakfast time. And
Oh, you know what I'm thinking about?
what are you thinking
The
about?
Vienna.
Vienna.
We
Yeah.
already talked about this, I think, though, in this course, didn't we?
Should we just go
I feel
to,
like we've talked about it here. We should do a lecture from there.
from Vienna?
Yeah.
Yeah. Oh, the best poached eggs. Okay, no, we're not going there. Okay. Yes. So we're gonna do the topic test on Friday. Please don't miss it. It's really important for you to wrap up this course and think about other things. So yeah, please write it on Friday. What else? Anything else that we want to say?
Yeah, they're just some confirmations that are going back and forth in the chat. They're
good.
flying back and forth about which date.
Oh, good.
Squirrel Project due
Okay.
this Friday, same day as the test and the IA project due. So all we have left for the course is the final topic test, the IA project from the seminars, and the Squirrel Life.
Yeah.
Yeah, so it is. That's kind of actually by design, that by this point in the course,
Done.
there is not a lot left to do.
Yeah.
Part of the reason we do that is because, and we did it before the pandemic, but the pandemic made it
More
apparent,
urgent.
more urgent and necessary that you want to make people's courses and their grades for it resilient to perturbation, personal, societal, whatever, but saving everything to the end is not a great design feature.
No.
And it doesn't reflect lots of real world things either. So
And it'll give you a couple of weeks to focus on other courses. Although I believe that there is a small announcement coming about the IA with respect to when it's due, similar to the one with the EPP, but I will leave that up to Dr. Ward Campbell, who is very much in charge of those things. So there you go. Okay. What?
some questions like we were talking about before about people in groups who are not contactable.
Yeah, so if you've had some struggles with your IA groups, first of all, totally normal. It's kind of why we do,
It's one of the things. It's
one of the reasons why we do group work. It's really important to learn how to be a good group member. And so all of that is taken into consideration. If you have any concerns, please just write to Dr. Ward Campbell, let them know. We'll be kind of, spot checking and writing to students to ask what they contributed to the IA. So, all of those things will be captured, and we will do our very best to ensure that there is an equitable recognition of contributions.
Yeah, I think that's very equitably expressed.
Yeah.
There's some in the chats that are flying back and forth. There's potentially, it seems, for Squirrel Life, most people are seeing due dates for the 10th. Somebody had seen one for the 3rd. If there is actually both, it's the 10th. It'll be the later one.
Yeah, don't worry
feel
too
stressed
much
about it. Yeah.
Don't feel stressed. Okay. In fact, Squirrel Life was meant to relieve stress.
Even for the squirrels.
Even for the squirrels. Maybe not for the squirrels. Okay.
Hold on.
The fish is just getting better, so I feel like we just need to just stall a little bit.
So we had old music.
Old.
I can give you an old movie recommendation if
Old
you haven't seen it.
music.
Fish Called Wanda.
Fish Called Wanda.
Very
Definitely very funny.
Like what is it? 40? It's a lot. It's old, but it's.
Speaking
it's very
of
good.
old movies, can I? Because I don't think we've actually properly done this yet.
What's that?
Can I please ask? How many of you, and I'll ask you to stamp on the left-hand margin of the slide, okay? So on the black part on the left-hand side of the slide, how many of you have seen The Princess Bride?
Like today?
No. And how many, and then on the right-hand side, how many have not yet seen The Princess Bride? This is The Princess Bride check-in. If you have not seen it...
Consider it homework.
Yo. Okay.
Have fun storming the castle.
You think they'll make it?
Not a chance.
Okay. So delightful for those that have not yet seen it. This
It's the perfect movie.
When you need a moment to bring you joy, when you are feeling like you just need a real mind break from start to finish, please watch The Princess Bride. I think it's on Netflix, right?
If it isn't, we'll find it.
We will find it and put it. Okay, if you write to us and say, I need a mental health break from for 2 hours away from something, we will send you a link to a Dropbox where you can watch The Princess Bride if you need it.
Disney Plus has it, and there is a father here who made the family watch it when he heard that you hadn't. That's great.
That's really good.
Questions about squirrels. Do you have to be a trail? Could it be your backyard? Backyard.
Yeah.
Yeah. There's probably trails there.
Yes. Okay. Wonderful.
I have a musical artist
Somebody
suggestion.
says, my name is Inigo.
Hello. My name is Inigo Montoya.
You killed my father. Prepare to die. So good.
And then Christopher Guest.
Sorry, it's just going to be all Princess Bride now. I'm sorry. But please watch this movie. I watched it with a friend of mine once who was not familiar with American films and watched this movie and then said, what's the next really good one? Wait, give me more. I feel like I will never see a better movie than this. It is a fantastic movie.
Life is a search for good art, including movies, but when you find a perfect expression, a perfect artistic expression, and in the integrative, consolidative, collaborative art that filmmaking is, Rob Reiner and all of those people, from Mark Knopfler doing the music to everyone else who is in front of it, it's just beautiful.
Beautiful.
And go follow Mandy Patinkin on Twitter because his son is filming, he and his partner, and they're adorable.
They're all good people, right?
Never Ending Story is also great. This is true.
Oh, that's a
Oh,
good
but
movie.
the horse.
Oh, I can't watch the horse scene.
Oh,
It's horrible. Okay.
I'll just think of the fist punch on the
Yeah. Okay. This is a spectacular slide. Thank you.
World's End. Like Edgar Wright, The World's End?
Whew.
Like in the Cornetto trilogy?
Amazing.
A+.
Well done. Okay. We're gonna have to delete. Like we're gonna have to.
Oh, we might have to
Can
talk
you
about
screen-
Shaun of the Dead.
Can you screen capture this?
Again?
Yeah.
It has changed, hasn't it?
It has. Okay. Wonderful.
Dig it.
Thank you so much,
Dig it,
everyone.
dig it,
The slide looks
dig it.
fantastic. Okay. We're gonna clear.
One of my favorite thing about Shaun of the Dead is that when his mother calls him Pickle. It's all right, Pickle.
It's all right, pickle. Okay, here we go. We are wrapping things up. So
Is that apparent?
yeah. We don't have much to say.
We actually
today.
no. We have a lot of physiology to talk about.
No,
today.
not really. But anyway.
No, okay, maybe not a lot, but two very cool things.
Okay, and let's do this first.
What's that? Oh yeah, quiz.
Yeah. Homework. The endotherm ectotherm challenge. Stamp for me, please. Or write, or do whatever it is that you want on the slide. You're better at figuring out what to do than I am. The mammoth and the ice fish. Which is an endotherm, ectotherm? How does all of that work? Oh, sorry. Good. Super. That's it exactly. Nice and simple, getting started. Okay, how about the ice fish? Good. Perfect. When it comes to temperature, is the mammoth a regulator or a conformer? Is the ice fish a regulator or a conformer? Excellent. Perfect. Good. When we're talking about the hemoglobin adaptation, or at least hemoglobin trait, or lack of hemoglobin trait, I just gave it away. So for the mammoth, sorry, I went to the second one.
I've done that
The
too.
trait discussed in class. So clearly, mammoth, hemoglobin, there we go. Perfect. Good. What is the biological level of organization of that trait? Verena's like, I'm writing it out. Good, you're right. Totally. Question mark, molecular? Yes, molecular. So same for the ice fish, right? Because it's the same thing, or lack of molecular, but still a molecular thing.
It's at the molecular level.
That's right. When we're talking about the hemoglobin and the time scale at which this is an adaptation to the cold, what is the time scale? Is it chronic? Is it acute?
Is it a cutie pie?
Is it a cute little molecule? Or is it evolutionary? Okay, a little bit of disagreement. We're like pumping it up. We're getting a little harder. This is fine.
See, no acutes though. So
No
that's great.
acute, so that's really good. I would argue that it is also not chronic.
Yeah.
Because first of all, for the ice fish, it doesn't have them. So it's not like that trait is changing state over time. It just doesn't have any. And then for the mammoth, I would also say that it's not chronic because it's not like, it's still a fixed state. Like it's, you know, the molecule is circulating in the body. It doesn't like increase in, you know, in upregulation of hemoglobin during cold. It doesn't change its state in the context of cold. It just is an adaptation to cold. So,
And some enzymatics or some proteins, that can happen clearly.
yeah.
Right?
Oh, totally.
But this is just not one of them.
Yeah. Yeah.
It's
yeah.
there.
It's there. Okay. The type of evolution or the mechanism that is involved in the mammoth and it's different in the ice fish situation or scenario with respect to hemoglobin. For the mammoth, what are we talking about? What's the type of evolution? Oh, and throwing out mutation. Mutation definitely got both of those started, so very well done.
Yep.
And Verina's like, yeah, I'm going to keep on writing. And man, your writing is like, do you have like a pen?
Yeah, I was just thinking about that. If you're writing with a mouse, I could not do that. I'd be like,
yeah. Super.
I see two.
Convergent.
Yes.
Well done. Good. That's a thing as well.
Yep.
Nice. Okay. Natural selection, adaptation. Super. Maladaptive. I love that. Bottleneck for sure. Yep. Right? Super
Nicely
good.
done.
Super good. Okay. Now, that brings us to the next question. Is it adaptive or maladaptive? You have answered that question as well. So maladaptive in the ice fish. And Nancy's got a pen too. Look at that. Okay. Okay. Is the adaptation the result of environmental constraint or facilitation? So constraint or facilitation. Nice. Caitlin, you're writing it out. You're going to write out like environmental too. Look at that. Good job. I feel like Caitlin's using a mouse, but I'm still like envious.
It's still much better. Even smaller, like more precise than I could do.
Way. Yeah. Okay. Good. Constraint allows for selection that leads to adaptation. Facilitation just basically can let your genes go wild. So well done. Now, the consequences for the species in the context of climate change. Let's just think of more immediate consequences with respect to this mutation, this adaptation. If we raise the temperature of the mammoth by a couple of degrees, what's going to happen? What's going to happen to the ice fish if we raise the temperature by a couple of degrees? Okay, Catherine's like, death. Michael's 3 X's. I can see it in the ice fish eyes, these little X's. Dead, Lily wrote. Good job. Yeah, like kind of immediate, like by suffocation.
Yep.
Which is amazing.
Yep.
Okay.
3 little fishes and they're all dead.
And, is it Nancy with the yellow pen, wrote adaptation or adapt? Yeah, it's possible. It's so possible that the mammoth might adapt because it's not as immediately life-threatening for a mammoth to experience a temperature increase or 2 than it would be for the ice fish.
There would be an opportunity. And in what we've looked at structurally and functionally, on the left column, on the right-hand column, rather, with the ice fish, you have the absence of a thing. On the left-hand, which is then harder to deal with, there's nothing to deal with. On the left-hand column, you have different isomers, different shapes, different propensities, different efficiencies. And there's a lot more space for potential evolutionary change to kind of whittle away at that if there's time.
Yeah.
Always with the evolutionary aspect, if there's time.
Yeah. How cool is that? I love the mammoth and the ice fish story. It's like one of my favorite things.
The mammoth and the ice fish, they set across the sea. Won't someone explain it? I don't think about it. 3?
I thought I had a good ending.
Are ice fish experiencing a rapid decline right now? Yes, they are in trouble. The temperatures are warming. Now, they're not going to experience nearly as quickly as you might think because the Southern Ocean is really good at isolating itself from the rest of the world, except it's starting to mix a lot more. So, yeah, the ice fish are in real trouble. Yes, sad face. I hear you. They are a freaky fish. I love them. Okay, and I hope you enjoyed learning about them alongside the mammoth, because to me it's just a beautiful story about hemoglobin, and anyway, I just think it's cool.
It's lovely. Now, you can imagine these kinds of things folding themselves into topic test materials where there might be other taxa on the top, other adaptations, like things we've talked about before, some other things adjacent to it, different strategies. And then this is kind of how a really helpful way to think about it and to work together as you prepare and study to quiz yourself and go, okay, I can think about it in all of these contexts. So think about it as the example and what kind of contexts have I had to think about. Yeah.
Nudge, nudge, wink, wink. Yes. Do you want one of those asynchronous study tool things? Because they're not like super popular. Do you want another one of those for this topic test? Yeah, a bunch of you. Like,
There's some
I
in the
imagine,
chat.
like, some of you are really active on them, so, yeah. Okay, good. We will put one together. It may be, like, an hour later than usual, but
Almost certainly.
we'll put it together. It will be available by the end of today. Okay, good. Okay.
All right, hold on.
Oh, what?
I've screen grabbed that once, but it, of course, has become more...
Yeah, it's delightful.
Yeah.
Thank you.
We love the participation.
We love the participation.
It'd be great to be sitting in the same room as you.
Right?
But,
Yeah.
And when we do that again...
We will also be on Zoom, right?
We're going to use Zoom.
We're going to use Zoom. If we're ever in person again, we're still going to use Zoom in the classroom.
I'm going to have to develop, like, shoulder straps as I wander around the class, and I'm definitely going to take a header down the stairs.
Do you think they'll buy us GoPros and fancy iPads?
Oh, with, like, a thing?
Yeah.
That would be awesome. Do I think they'll buy it? No.
No.
Could it be purchased? Yes.
Yeah. Okay. Okay, here's a polling question, and the answers were way too long to fit in the polls, so you have to pick between A, B, C, or D, or E as the last polling question. There you go. Please don't write on the slide.
Oh.
And,
Please don't write on the slide.
well, don't write on the answers anyway.
Yeah.
You can write anywhere else. It's fine. But just don't share what you think the answer is.
Come on, get up. I want to take you.
What?
I'm just...
Good noise?
No.
Good news?
Frustrated noise.
Oh. Look.
Important. That's great news. Should we celebrate it?
It's worth celebrating.
Mm-hmm.
Oh, boy.
We have almost cracked the 60% participation.
Yep. Come on, get your answers in, and we will move on. I think we won't wait until we get to 90, though. Not today.
No. we
They're
got stuff
tired.
to do.
Yeah, they're tired. That's okay. So, we have good news, and also wanting to wish all those who celebrate a very happy Hanukkah. Last night was the first night of many. I remember when I was in university, I wasn't allowed to light candles because they were worried about it being a fire hazard. And it was in a city, like I must have been the only Jew for like 800 miles. And so they had no idea what this was.
Where was it?
I'm not going to say. Because I really do hope that the Jews have moved in to the city.
Were there... Can I ask if there were already Roman Catholics there? Because they light fires all the time. They have like a bank of fires in an old, dusty room.
This was not well received. Anyway, yeah. Okay. I'm gonna stop it because we are saturated now at 77%. Oh, it's 78%. Okay.
Super.
And poll. Most of you did get the correct answer though, so that's really good. So the answer is C. Non-adaptive traits can still evolve in the context of environmental facilitation. This has important restrictions in the species' resilience with respect to climate change. Very good. All of the other ones are not true for some reason. So what we want you to do, of course, is to go out and do two things. Tell us why it is not true, and also make it true. So you can do that in a bunch of ways, right? Okay. Good. Oh, sorry. This is like the last time that we get to see you or be with you. So, one of you wrote to me, thank you for writing, to ask about the EPP and the number of spots on the insects, because sometimes it was difficult to tell exactly how many spots, and that was the point. Yay! Okay, so biological samples, sometimes not always super easy.
Literally sometimes missing the part of the body.
And sometimes they're blue and yellow, like so those things are introduced in order to get you to ask exactly the questions that you're asking. We are not really interested in whether or not you got exactly the same number of spots as we did. What we are interested in, rather, is how you handle the data, how you move it from one sort of section of the scientific method to another, how you make decisions, what you evaluate, right? And so I hope, anyway, from being in our class and our course, that is a little bit more comfortable. It shouldn't be perfectly comfortable yet, but we're just kind of trying to recreate some of those moments of uncertainty to kind of provide you with the opportunity to make decisions and be confident in them. And the confidence comes later, and even now still.
And if you're still looking for more concrete suggestions, your colleagues in the chat have thrown up a couple of great ones. And that
Great.
also is kind of analogous to what happens in the real world when you start to measure the biological variability, that some of it's about species variability, some of it's about just the preservation or the age or the sex or all of these other, the angle the camera is at.
Yeah.
There's tricks in you, so you kind of learn from the people who are doing the same things.
That's right.
As you.
And what we learn, especially in ecology, is that you don't necessarily have to be 100% accurate. You just need to be 100% inaccurate. That means that if you do something to sort of overcome some barrier, that you are consistently inaccurate, right? Because you can compare things if you are consistent. And accuracy is something we all just aspire to, more or less.
Yeah, precision. And if you can be precise,
Yeah.
you can...
Yeah, cool.
Yeah.
That's a total sidebar.
Yeah, we're going to show it as a... there's a question I just want to call. Somebody's asked about a quick explanation about facilitation versus constraint. Do you want to keep moving in this and talk about that in the office hours, or do you have a quick one?
Okay. Facilitation versus constraint. So just trying to tie in our evolutionary processes into ecology. And what we want to do is suggest that sometimes the environment presents limitations and challenges to species, and sometimes it doesn't. And that's kind of what we mean by constraint, so challenges, versus facilitation, not presenting challenges. And so when the environment presents challenges, right, physiological challenges or otherwise, that provides the necessary conditions for mutations to be selected for and adaptations to evolve. Okay, so it's kind of taking that whole mutation, adaptation, kind of natural selection thing and adding now the environmental context under which that could happen. Facilitation is the opposite of that. It means that the environment is so wonderful and cushy and comfortable that it doesn't present, at least in that context, some kind of challenge. So there's no selection, which means you have the potential for the evolution of maladaptive traits. So that's what we were meaning by that. Okay.
We should keep going.
We should keep going. This is covered in some of the course link stuff, but we thought we would talk about it because it's about fat, and I love it. Okay, yeah, it's so good. This to me is like the ultimate, like, in amazing. Okay, so the homeoviscous adaptation. This is in ectotherms. What?
I just think it's a great name for a band too. Live tonight.
And ectotherms, where the composition of the phospholipid bilayer can change. So there's all sorts of different types of fat, you've heard about good fat and bad fat. That's also like, the good fat, bad fat thing is more about how they're constructed, but also the actual difference in the molecules, right? So there's like dozens and dozens of different types of fat molecules, and they all vary by the length of the carbon chain and the number of double bonds within it, okay? So like fatty acid 22,1 would be 22 carbons long with 1 double bond. Fatty acid 22,0 would be 22 carbons long with 0 double bonds. That would be a saturated. An unsaturated has at least 1 double bond, okay? Now, the double bond thing, and this is why knowing a little bit of chemistry is important, a little bit of physics, but again, like you can always learn this stuff on the side. But the double bond thing is really cool because if you think about what it does physically to the molecule,
Then it all makes sense.
it all makes sense, okay? So here's what we've got. Stand up.
Me?
Yep.
No, forget it.
No.
Forget it.
Stand up.
Oh, yes, Ann. No. No. No.
Okay, so imagine that Smith and I are 2 phospholipids in a fatty acid bilayer, okay? We're 2 phospholipids, right? And if we're saturated, it means we have no double bonds, right? So we're just side to side like this, okay? And if we're all, like the whole layer is like that, it's kind of very rigid, right? They're all kind of stuck together. But if you have at least 1 double bond, what happens is it kinks the molecule out like this, okay? And it has all of this space. Look, there's space, okay? You get a little kinky too. There we go. Okay, so it creates like a physical space. The kinky molecules occupy more space than the totally straight ones, right? Okay, so, are you good?
I'm great.
Okay.
I would have warmed up.
Okay, so the space makes the bilayer more fluid, right? It increases the fluidity of it. And this is really important when we're talking about sort of getting molecules through membranes in the cold, for example. The more sort of packed together the fatty acids are, the more that kind of freezes up. It loses its viscosity. Its viscosity changes, and so what we want is things that are more fluid, more kinky, right? So that, you know, things can pass through them. So, so anyway, fatty acids are amazing. So we want to have more double bonds in your phospholipid layer. And what can happen, like, so this stuff is like really cool. So, cholesterol, for example, is like this monster fat globule that kind of gets into your bilayers, your lipid bilayers, and increases the rigidity of your membranes. That's one of the reasons why cholesterol is super bad. It makes things rigid, and reduces this fluidity of the different membranes. Too much fluidity is bad. If you heat up, for example, you can denature the lipid bilayer, and then it falls apart, right? And your cell dies.
And you said you would want,
Yeah.
i.e. they would want, i.e.
Well,
in the context of being in
evolution.
the north,
Yeah.
right?
Yeah.
In the cold.
In the cold.
This is
More
in the context
phospholipids.
of being in the cold.
That's right. So more double bonded phospholipids in the bilayer is the homeoviscous adaptation. Okay. But then, let's bring this up a notch, another level of organization, because I used to get this question all the time. So the question used to be, when I was traveling in Antarctica, people would say, okay, so why don't penguin feet freeze? And this is an endotherm. This is not an ectotherm with the homeoviscous adaptation. This is bringing us up a level of organization and also moving us over a taxonomic group, or 2 or 3. And so they would say all the time, why don't penguin feet freeze? And it's kind of an obvious question. It's not a bad question. The better question is, why don't penguins freeze? Okay? Because penguin feet are very nearly frozen. this is another adaptation to living in the cold. So, regional heterothermy.
Another good name for a band.
Oh.
On a double bill with the viscous...
Homeoviscous adaptations?
Yeah.
Ha! Okay.
Who would open for who, though?
Right? Okay. So, regional heterothermy isn't about a molecular...
So I just want to throw in, I think this is more cool than the fat.
Oh.
Now, you have a fat bias. You study fat.
Oh.
I think this is the thing that I would aspire to. If I could do this, we could paddle. Oh my God. We could just paddle all the time.
The reason why we have these kind of stuck together is because both of them are cool, but also because they really speak to the idea of form leading to function. Okay, so the form of the molecule leads to the function of fluidity in the membrane, right? The form of countercurrent systems lead to the function of heat exchange. So what we have is, instead of just veins and arteries that go up and down into the feet, what we have instead is this complicated web, this net that takes the veins around the arteries and creates a heat exchange system because they are in such close proximity. So as we move further and further down the leg of the penguin, it turns out that it gets colder and colder. The body temperature in that area is colder and colder. And that's why we call it regional heterothermy, right? Regional, just meaning in an area. Hetero, right? Meaning multiple or different. Thermy, temperature. So the body has different temperatures depending upon the region, okay? And it turns out that a penguin's leg is not super tiny. That's like another thing if you want your mind blown. The penguin's leg actually starts where other legs start. It's not that they've adapted to be smaller, okay? So if we take a penguin, like this one.
Here.
Thank you. We take a penguin, like this one. Its leg doesn't start, like its knee isn't even there. Its knee is up inside its body, okay? And its hip is up here, right? So this is the leg of a penguin, more or less. Okay.
So they wear inverse old man trousers.
They wear pencil skirts. Okay. Penguins wear pencil skirts. Their whole like fat and body kind of like come around down to their ankles because it's freaking cold where they live.
And that's why they can't pick up things they drop on the floor.
Yes, so their bodies and their guts basically come around here and keep their big thigh muscles, these big thigh muscles, nice and warm,
yummy
right?
Drumstick.
Yeah.
Yummy drumstick.
Rather than being exposed to the cold, they're covered in this layer of fat. And then by the time they get to their feet, there's like almost no muscle. Like it's just tendons and wiry and really cold. Only a couple of degrees colder than the substrate. But that's what they do. So the warm temperature, the warm core temperature goes down, but then it gets transitioned over to the veins coming back, right? Keeping the feet very, very cold and keeping the heat within that sort of pencil skirt of the penguin. Penguins have long legs. There you go. Okay. So this is kind of what it looks like. We have the veins and the arteries in very close proximity. You can see the temperature going from 32 degrees Celsius. And in fact, the core temperature of a bird is like 39. But then it comes down and there's heat exchange that lowers the actual temperature because the heat isn't going down with the artery. It's going across up with the vein. Okay, so that prevents the core of the bird from cooling down. And keeping that high body temperature, the countercurrent heat exchange system. Kind of cool, right?
I think countercurrent heat exchange is pretty amazing.
Yeah.
Yep.
Okay. Or you can wear a sweater. and so this is a delightful fundraising thing. You should definitely, if you can, give money to Saving Penguins. You don't need to knit them sweaters, though. But anyway, what's this?
Well, this is kind of a cool thing. This is kind of what happens to us. So this is a picture of me paddling when it's minus 20 out, and the only reason there's no ice there is because it's quite a fast flowing river. And so you'd experience this. So we don't have a countercurrent heat exchange, of course. Would that it were so simple. But what we have, what people have, what some other mammals have is what's called cold-induced vasodilation. And where do you see this a lot? You see it in people who work in fisheries, on ships. where they experience this drop. So temperature in your fingers is the y-axis, and then time of exposure to the cold. And you've experienced this, whether or not you've worked on the tuna ships or you've been out in the cold in the wintertime. And so you've experienced this where you know that when you go outside, there's a drop. Your body protects the core by saying there is something cold here at its extremities. Take it away. And so your temperature drops. And I think you also know if you stay outside, you're going to see it return. And there's a delicious return of heat after 5 to 10 minutes after your exposure, if you stay out there and stay moving, and then it will periodically begin to ramp up and down through. So protecting the core from the cold that would be the cold blood that would be returning from its extremities, but also kind of sending it out to maintain the extremities. And you can see why this is a strategy in terms of the acute chronic thing. This is only a useful strategy if it's an acute stress.
Right.
That if it's something that you're exposed to for relatively short periods of time. So I would paddle for several hours, and then I would go in and I would enjoy soup.
Yeah.
And soup is hardly ever available to the penguins of Antarctica.
Right.
So they need a different strategy.
Yes. Lots of different strategies. So check this out. We can kind of go up and down. Remember, why is the background green?
It's because we can put in whatever we want.
It's the Southern Lights. Okay. So what we'll do is we'll take you like across biological levels of organization, and we'll talk about sort of adaptations to the cold for the penguin, right? So at the population level, and not everything has adaptations at the population level, but penguins do. At the population level, penguins huddle together in order to maintain body heat, right? Some penguins, not all of them, the big ones, especially the ones that breed during the winter in Antarctica, because it's cold, okay? At the organism level, there are all sorts of adaptations. So an emperor penguin, for example, has a relatively shorter bill than a king penguin, which is basically the same, a little shorter, and has a much longer bill because they live in relatively warmer conditions. It's still not warm.
Compare it, yeah.
Yeah. The emperor penguin have shorter fins, right? So they're reducing their appendage size, and they are big and blubbery and full of fat. They are rounder than most penguins.
Somebody asked actually in the chat about what they've all seen in terms of penguins incubating their eggs.
Yeah.
Why is that on the coldest thing? And then you're settling all that big blubbery. It's settling your pencil skirt of
the pencil
fat
skirt
and
goes
warmth.
up around underneath, right?
Yeah.
And in fact, they have a brood patch. So all birds. I'm going to say all birds, and like
All
some
birds?
of you who know birds are going to be
Many?
like, what about the? And you'll be totally right. But all birds have brood patches, which are these bare areas on their bellies where they don't have any feathers that put the skin in direct contact with the egg, right? And then they kind of wrap their feathers around, and it's like basically bringing the egg back inside of you without doing it.
It's a snuggle patch.
It's a snuggle patch. There you go. Okay, so they have those. So they use their feet basically just as a holder to keep it dry as well, and then they cover it with not only their brood patch but also their feathered gut. And that's how they keep things nice and warm. Most penguins have 2 eggs. The ones that put them on their feet only have 1. Okay. And then finally, of course, at the organ system level, we talked about regional heterothermy and the way that is accomplished by this like intimate relationship between the veins and the arteries in order to be able to pass heat to preserve the temperature of the core. So, here are some things, right?
Let's read them out loud.
No,
let's not.
But remember, we gave you this table for the polar bear, and we asked you to do a very similar thing. So we've done a thing for penguins that you may want to use as kind of like a sample of what you might want to think about for the polar bear. This is just us going through the literature, trying to figure out where each of these types of adaptations might exist, right? And of course, there will be some blank squares. And so we could ask you, for example, what some of the possible interpretations are of the blank squares, right? There are multiple hypotheses that could explain a blank square. So think about those reasons. Why? Okay. Let's wrap it up.
All right.
Okay?
With a bow.
With a bow. So, we talked about this already. Where in the world are you going to see the greatest change in average temperature? Go ahead and stamp for us. Yep.
I'm glad to see some southern hypotheses.
Yep. Really good. That's great. Okay. So we're going to clear?
Yep, got it.
Got it?
Thank you.
Okay, moving on. Where in the world will we see the greatest decrease in alpha diversity? Decrease in alpha diversity.
Yeah. Yeah.
Well done. Yep. Super good. Yep. That's great. You got that one?
I do.
Okay.
Thank you.
Thank you.
No, thank you.
Where in the world are we going to see the greatest increase in alpha diversity?
Slightly.
Yep. I do like that most of you are ignoring Antarctica, because in this case, it would be hard for things to get there. There would be a slight increase, but certainly the greatest increase is going to be in the Arctic. Well done. Okay, you got that?
Got it.
And if you remember, we asked you before, on a scale of 1 to 10, what do you think is going to happen to the ecosystem? Most of you said D, right? Dramatic changes, migration of some species, invasion of some southern species, overall decrease in biodiversity. Remember, we all said that? We all think that. What you've just told us, though, is that's not actually one of the things that's probably going to happen, which means we learned some stuff, which is amazing, because there will be an overall increase in biodiversity, or at least as measured by alpha diversity. Now, it could definitely be a decrease in Arctic adapted biodiversity, for sure, right? And the endemic species. But the overall decrease in biodiversity is not likely to happen anytime soon in the Arctic. And yet, if somebody says an increase in biodiversity, most of us would be like, oh, that's a good thing. Is it? So we need to take a look at it from a different perspective, right? So regional temperatures are increasing really dramatically, especially in the Arctic, for all of the biogeographical reasons that we've talked about. And instead of talking about alpha, we probably should be talking about beta diversity, right? And that's kind of your favorite.
I love it. I love it. And we should probably, why should we be talking about beta diversity? Well, remember, step back with me. Remember, previously on Biology 1070, when we talked about alpha diversity versus beta diversity, the beta diversity is the similarity or the dissimilarity between two areas or two time periods. And so the beta diversity that we ought to be concerned about is how dissimilar things used to be, perhaps compared to how similar they are becoming, how homogenized they might become. Who's arriving, who's leaving, and how similar or dissimilar things are in 50 years, in 10 years, compared to contemporarily. And I also love to throw in, because you identified with those, where is the greatest physical change happening? It's in the north. Where is the greatest change in alpha diversity? It's in the central latitudes around the equator. But here in this slide, we remind you that where does that change happen because of the same abiotic factors that we talked about in the north, but also strongly affect biodiversity? High elevations. So the Arctic and the high elevation. So the Arctic is being hammered with these greatest changes, physical changes, and the high elevation, particularly in the tropic, those places have high diversity, and they are being hammered by the highest changes in temperature. So this turnover that we're expecting to see through time we're only able to measure that if we know where things are now. And so this is I suppose, my invocation to you to keep becoming biodiversity scientists, because as awful as things are, they could still be worse if we don't change them, and they can still be ameliorated by better understanding and more action.
And knowing a little bit more about the diversity of life may invoke a little bit more humility in all of us. So
I think it ought to.
Yep.
That's certainly what it does to me.
Yep.
Yeah.
Some
This is just an
playlists.
aside. There's all of, if you wish to go through this whole thing again, you can watch all of the classes start to finish. Or if you liked any of the music that was being played beforehand, that's all there too. And there's more stuff we didn't get to because we had to choose only 22. And we screwed up once and played some, I forget what track it was, but we played one track twice.
Oh, so yeah, study playlist available to you as you prepare. Folks, that's all. And we are so grateful to all of you for sticking with us, for participating, for being awesome. And it's really quite a privilege for us to be able to have this platform to share our ideas and our opinions and our science with you, and certainly recognizing that privilege. We have talked at you for 22 hours, and hopefully you've been able to share some of your personality and thoughts and opinions with us in some way. We will be here on Wednesday, but we will not be teaching. We will be doing whatever it is that you ask us to do. So come prepared. Yeah, come prepared with questions if you want. But we won't be presenting any new material or anything like that. Giving you a little bit more time to prepare for the topic test on Friday. Okay.
Yeah, and I know we're over time, but I just thank you and continue to insist for yourselves that you deserve to receive kindness.
Yep.
This shit is not over, and you're persevering in spite of it.
Yeah.
And
Amazing.
so well done you.
Thank you. Okay.
Receive it, deliver it.
Okay. Bye. Screenshot it. Screenshot it.
I'm going to make a video of it too.
It's lovely. Good semester.
I had to remember why I put that slide in
Oh yeah.
2 years ago or a year ago. What does it say in yellow? Nice. So we're not muted?
Well, we are muted.
Oh,
but
I see.
They can close caption us.
Lots of nice comments.
Mm-hmm. Hey, Gracie.
What? That's good. So I'm a little bit.
Yeah.
Uh,
A little bit not muted.